Hello everyone. Till now we have studied about recombinant DNA technology, and now we will be looking at the techniques which are very important in the recombinant DNA technology and as well as in whole biological experiments. First, in this we'll first be studying about gel electrophoresis. Gel electrophoresis is nothing but method of separation of different fragments of the DNA or RNA on a gel medium. And next is the cloning libraries. Cloning libraries will include all the identified cloning sites. And then restriction enzyme mapping is also very important for knowing for identifying the parts of DNA where the restriction enzymes will make cuts. Polymerase chain reaction is a reaction for amplification of the gene. Then nucleic acid hybridization is a method in which hybridization of the DNA with labeled probes is being done to get the gene of interest. Then is the DNA microarray. DNA microarray is the process of it is a process to know the expression of many genes in one go. Now let's start. Gel electrophoresis. Here it has been shown gel electrophoresis. The gel is taken. This gel can be of agarose or polyacrylamide, and here the wells are made. In the wells the sample is added, and in the presence of buffer electricity is applied. By applying the electricity, that the different lengths or fragments of DNA can be separated, and the buffers used in this electrophoresis is TAE or TBE. TAE stands for tris acetate editor and TBE stands for tris borate editor. EDTA stands for ethylene diamide tetraacetic acid. It is also known as sieving effect, means it separates the different sized DNA fragment, and the DNA is negative charge due to the presence of phosphate group in it, so it will move towards positive electrode, that is anode. Cloning libraries. In the cloning libraries, different clones are collected. The collection of the DNA probe in a certain vector is done in this cloning libraries, and each gene is mentioned in the libraries at least once. It will be used, its presence can be seen by the cloning libraries. Genomic DNA made from the restriction enzyme DNA. The genomic DNA is made by restriction enzyme DNA, and the complementary cDNA is made from the mRNA by reverse transcription. It is just basic information. Now restriction enzyme mapping. Restriction enzyme mapping sites are mapped for further manipulation of the gene. First, the digestion of the gene is done singly and then in combination with several enzymes. The fragments are subjected to gel electrophoresis to separate the fragment by the size, and the sites are deduced on the basis of size of fragment. And as earlier we have studied about the restriction enzymes, they make cut at specific sites of the DNA and create sticky ends, and these sticky ends will be further ligated with the gene of interest. The DNA of the host would be ligated with the gene of interest because it has been cut by the same restriction enzyme. Coming to polymerase chain reaction. Polymerase chain reaction is a method of amplification of specific fragment or segment of DNA using a probe. It was developed by Carey Mullis. A very small fragment of DNA can be amplified to large amount. Only 30 cycles can produce about 1 million copies. It is carried out in 3 steps. First step is denaturation, second step is annealing, and third step is extension. In denaturation, high temperature is provided and both the strands of DNA get separated into 2 separate strands, whereas in annealing, the 2 strands, at the ends the probes are been added, which will help in making the complete double stranded DNA. Extension, this chain, the probes would extend to form complete DNA, and this cycle will be repeated multiple times to get the amplified DNA or RNA. The instrument, which is thermal cycler, regulate the temperatures difference in all the 3 steps. Now coming to nucleic acid hybridization. It is a method of obtaining gene of interest by probing DNA fragment which have been separated by electrophoresis as labeled probe. Southern blotting is used for identifying specific DNA fragment by using the DNA probe, whereas northern blotting is used to identify specific RNA fragment by using DNA probe, and western blotting is used to separate amino acids from the protein. Here maybe it is southern or northern or western blotting. All these techniques are working on the same principle. A bit difference is found in the western blotting, where the probe where we use primary and secondary antibodies, and it is a bit complicated, but southern and northern blotting are all similar. First we take the sample, then we will carry out electrophoresis. The sample will be either DNA or RNA fragment. Then the electrophoresis will separate the DNA or RNA fragments according to its size. Then the segments has to be transferred to the nitrocellulose paper, and then by capillary action it will be transferred to the nitrocellulose paper, and after putting the labeled probes will find its complementary DNA or RNA, and when exposed to the UV rays it will be easily visible and we can get the gene of interest. Now coming to southern blotting. As explained, in southern blotting we will take the DNA, and on the gel, agarose gel, will provide electricity, and in the buffer the electrophoresis will be carried out, and after that DNA fragments will be separated, and then these separated fragments has to be imprinted on the nitrocellulose membrane, and this is done by capillary action and the presence of salt solution. And after this nitrocellulose, which has the DNA fragments on it, the labeled probes are added to it, and the labeled probes finding the complementary sequence will hybridize, and when visualized by X-ray exposure would be visible. It was developed by E.M. Southern in 1875. The similar is process for northern blotting. Northern blotting also RNA is taken and then electrophoresis is carried out, and then it is the RNA fragments are separated, are transferred to the electrophoresis is carried out. But in electrophoresis we also add formaldehyde agarose gel electrophoresis, because formaldehyde, as this is single stranded and in this strand only complementary pairs are present, so the RNA becomes twisted and find its complementary base pairs within itself. So for preventing that we use formaldehyde, and then after electrophoresis the DNA fragments is transferred to the nitrocellulose membrane, and then we add the labeled probes. After adding the labeled probes, after application of after visualizing it under the X-ray, it will be easily visible. This Nathan dotting technique was performed by Ovkine and his colleagues in 1979. These are the difference between southern and western blotting. Main difference lies that the southern blotting and the northern blotting both are carried out on the agarose gel, but in the southern blotting just agarose gel. In the northern blotting we use formaldehyde agarose gel, and in western blotting it is SDS page, means sodium deodecyl sulfate polyacrylamide gel electrophoresis. And DNA microarray. DNA microarray is very interesting technique, and it is used to detect the cancer in a cell. We take the sample in which the normal cell sample and the disease cell samples RNA is isolated, and then from this RNA we find the complementary DNA by reverse transcription, and then the label probes are added. By adding the label probes, the label probes will hybridize with a segment of complementary DNA, and then in the plate, microarray plate, this all samples are kept. When visualized under the microarray scanner, it will show different colors, and the color will indicate how much the cell is affected, whether the cell will be gene is activated or not non-activated, whether it is activated in both disease and normal condition, or whether it is just active in the disease condition, or whether active just in the normal condition. Now coming to applications of recombinant DNA technology. Recombinant DNA technology has wide field of applications, being it therapeutic products, genetically modified products, diagnosis, or energy applications. In the therapeutic products, we can find its application in the vaccine making and growth hormones, making antibodies, vectors, recombinant proteins, anti-cancerous drugs. In genetically modified products, we can see its application in genetically modified fruits, vegetables, crops, microbes, and these all edible fruits or vegetables are more energy giving and having more shelf life too, and the quantity and quality both are increased. And then in the diagnosis, in the gene therapy to treat adenosine the aminase deficiency disease, CRISPR in monitoring devices and therapeutic strategies, and in energy applications, biohydrogen, bioethanol, biomethanol, and biobutanol, means in getting efficient energy which is very environment friendly. This was all about the recombinant DNA technology and the techniques used in this recombinant DNA technology. Thank you.